for being here, especially under these really trying times. I'm sure many of us did not get a good sleep, but hopefully this time can serve as a way to redirect our attention to celebrate the incredible work and creativity of the Black and Neuro community. We'd like to thank our sponsors up on the screen here for making this event possible. I'd also want to give a huge shout out to the Black and Neuro team behind the scenes who are making sure the logistics run smoothly for this event. It is quite a feat to be doing. And in particular, I want to say a huge thank you to one of our team members, Monica, who has worked tirelessly to make sure that the organization and the logistics for these talks specifically have run smoothly. So thank you to the Black and Neuro team. Now, for a few logistics on how session 3 of the Data Blitz talks will go. You, up the bottom of your screen, on the Zoom screen, have access to live transcripts. So if this is something that you need, please feel free to click on that and the transcript will appear at the bottom of your screen. Actually, you can choose where it will appear. And just feel free to DM us if you're having any issues with that. I will also ask if all participants can rename themselves if they haven't already with their first and their last names. That way we can find you easily when it is your time to speak. With that, we will also be messaging you if you haven't already. So when it is your time to speak, you will know that you just have to turn on your video camera and go ahead and start talking. Of course, unmute yourself as well. I will be moving the slides, so just let me know when you would like to move on to the next slide by indicating or saying next. Now, here we go. I would just also like to ask our audience if you have any questions throughout our speakers' talks. Please feel free to drop them in the Q&A box. And please make sure that you are adding the speaker's name to this question so we know who to direct the questions to at the end of each session. And right over there on the chat, yes, so there's the live transcript button. Okay, so everyone knows that. Again, please DM us if you have any logistical issues. We're going to get started with the first session. And before we do, we'd like to thank our judges who have taken their time to be here and to provide feedback for this session. So specifically, thank you to Dr. Cristina Rodriguez, who is a postdoc in the G Lab at UC Berkeley, working on advanced microscopy methods for imaging the brain and spinal cord at depth. And also to Manuel Moore, who is a postdoc in Dr. Chen's lab at Stanford, where he applies his background and passion for molecular tool development and imaging to neural circuit investigations in the brain. So thank you both for being here. So as I mentioned, this session 3 will begin with talks on various topics from sensory and motor systems to neuroengineering, methodology and tool development, machine learning, and computational neuroscience. So let's get started. And we are going to get Kendi Cole to present her selected talk. Sorry for butchering your first name there, but feel free to begin.
Thank you. Hello, everyone. My name is Kendi Cole, and I'm a fourth-year PhD candidate at the Integrative Neuroscience program at Stony Brook University, and I work in the Parsons Lab. Thank you for giving me this time to talk to you about a couple of works that I have been doing in the past year, and it's still ongoing, specifically looking at how prior experience has the capacity to alter future learning. Next slide, please. When it comes to the study of learning and memory, significant progress has been made in identifying the cellular and molecular mechanisms that support the acquisition and consolidation of long-term memory. However, One area in learning and memory that's not very well understood is how prior experience affects subsequent learning. And this is due to the fact that most of the studies on learning come from studies where the learning event is usually presented in isolation of things that happened prior. And I think we can make a pretty obvious case that understanding what happens prior before an experience is evident and important in understanding how memory develops. But with that being said, there have been a handful of studies, in vitro studies, that have been able to show that the ability of cells to express long-term synaptic changes can be influenced by their past history of stimulation. And this is one of such studies from 1996 from Cohen and Abraham. Here they looked at the influence of prior metabotropic glutamate activation on subsequent long-term potentiation induction. So briefly, what they did is they took recordings of cells and they got like a good baseline. And then afterwards, they baited cells in this priming agent, ACPD, and it caused a depression in the synaptic responses. And after the priming agent was washed out, the synaptic responses basically went back to baseline. And then 30 minutes after, they induced long-term potentiation via theta bus stimulation. And this resulted in an enhanced magnitude and stability of long-term potentiation when compared to the control group. So basically what they were able to show is that there was a change in neural function as a result of the prior pharmacological activation that persisted after the washout of the prior stimulus, and that this altered the response to subsequent long-term potentiation induction. Next slide, please. But subsequently, there have been analogous studies in behaving animals also looking at this phenomenon on how prior experience is able to alter future learning. And this is an experiment that my PI did in 2012. We published this paper where basically using a unique two-trial fair learning paradigm, here we're able to show that when an animal is presented with a weak event, it only produces short-term memory, but it does not support long-term memory. But if this event is followed by an identical event, either 60 minutes or 3 days apart, what you get is robust long-term memory formation. So what we are seeing here is that this first training trial engages a priming process, and this process is active such that when you deliver, next slide, please. When you deliver a second training trial, next slide, please. You deliver a second training trial when this priming process is still active. Next slide, please. What you get is long-term memory formation. And we're calling this event here priming because the ability of this first week trial to prime subsequent learning eventually dies out, such that if another trial is given either too close, next slide, please, or given too late, next slide, please, that is about 30 days after, you don't have any significant effect. So next slide, please. In the next couple of experiments, what we're now trying to do is investigate the role of the amygdala in this priming effect. Reason being that one of the fundamental principles of fear learning is the necessity for activity-dependent plasticity within the amygdala. Briefly, the amygdala circuitry mediating fear learning is already well established, and major brain areas include the amygdala. The lateral amygdala receives converging input about the stimulus from the thalamus and the cortical areas. And one of the other important areas is the middle portion of the amygdala that sends outputs, the target areas. And one of the things that we measure in my lab using the fear conditioning paradigm as a measure of fear is freezing behavior and increased startle. And so there have been hundreds of studies that have investigated and shown the importance of the amygdala circuit in conditioned fear. And as such, we are now trying to determine the role of the amygdala in this priming effect that we see. Next slide, please. So one of the questions we're asking is, one, if we simply block activity in the amygdala, do we prevent this priming mechanism? Next slide, please. Secondly, we're also asking whether neural activity and cell signaling events in the amygdala change as a function of the time following that initial priming event. Next slide, please. So in order to do this, we use a chemogenetic technique known as DRETS. In the past couple of days. A lot of people have been talking about dreads. So briefly, without trying to go into that much details, what this technique allows us to do is allows us to be able to modulate neural activity by this mutated dread virus, and we put it into the brain region of interest. And depending on the kind of receptor you're using, either inhibitory or excitatory receptor, we are able to control neural activity that way. And these receptors, like I said, since they're mutated, they are not they don't respond to endogenous ligands found in the brain, like acetylcholine. And here's just like pilot image just showing you expression of the virus in the amygdala. Next slide, please. So the first question you're asking is whether if we blocked activity in the amygdala, would we prevent this priming mechanism? And so we gave the inhibitory dread virus in the animals, and after 6 weeks, we gave them 2 trial fear conditioning paradigm separated by 24 hours. But what's important here is we gave that injection of CNO, which activates the dread receptor right before that priming event. And what we see here is that a pre-training inhibition of the amygdala was able to prevent subsequent long-term memory formation. And this is very important because one of the things that this experiment tells us is that the amygdala is basically important. And here we just have like different data that we collected just here, basically showing that the animals were able to respond to the shock, and that the deficit you see here is not by virtue of the animals not responding to the shock. And here is just to look at off-target effects of clozapine N oxide. So one thing we know is that the amygdala is important, but what we don't know is what it is about inhibiting the amygdala that basically prevents this priming process from happening. We also know that plasticity markers in the amygdala are active 1 hour after a single training trial, and that pre-training inhibition disrupts this process. So next slide, please. In the next experiment, what we are now trying to determine is if disrupting neural activity post-training would have any effect on memory consolidation, thereby preventing subsequent memory. So here, the experimental paradigm is still the same, but one of the most important things that's different from the first experiment is that we gave the CNO injection right after that first training trial. And what we see here is that post-training inhibition of the amygdala had no effect on memory consolidation. So based on the pattern of findings from both experiments where we see a pre-training inhibition but not a post-training inhibition, it might be that whatever mechanisms that allows for the priming process to occur basically happens very rapidly, that even if we give CNO post-training, it seems to have no effect. It could also be that our dread manipulations are not affecting biological processes that still occur during that first training trial, and that these processes are still happening, and despite the inhibition, that they might be dependent on cell signaling molecules like PKA or MAPK. And so using drugs that have a well-established effect on post-consolidation effect, we now set out to determine if disrupting neural activity post-training using a pharmacological inhibition would prevent subsequent memory formation. Next slide, please. Okay, and so what we did here is we basically had the learning paradigm the same, but here we're trying to block activities of the cell signaling molecules that I talked about, MAPK and PKA, using RP-CAMs for PKA and U0126 for MAPK. And what we actually find here is that a post-training blockade of MAPK or PKA does not affect long-term memory priming. Next slide, please. So basically, the present results show that the ability of an initial weak fear conditioning paradigm to alter future depends on rapidly developing neural mechanisms, because we show that inhibiting the amygdala before but not after initial training trial prevents that trial from facilitating memory for a second trial when tested 24 hours after. Collectively, our results indicate that the ability of an initial fear conditioning trial to facilitate subsequent learning depends on neural mechanisms that develop rapidly during the initial trial, and that the fact that we also did not observe a disruption when we used PK or MAPK inhibitors after the trial, the first training trial, lends further support to the conclusion that the memory priming effect of the first trial does not have a typical consolidation process like we've seen in some other studies, but I didn't show them here. Yeah, thank you. So next slide, please. I just want to thank everybody in my lab, from my PI to my RAs and all the members in our lab, those that are here and those that are already left. Thank you.
Thank you so much for that talk. So we're now going to invite members of the audience to submit any questions that they may have. So please feel free to use the Q&A button, and we will have a few minutes to ask them. Okay, so we have one question here from Julie. Great talk. If not MAPK and PK, what signaling process do you think are regulating this?
Thank you, Julie. That's actually a very interesting question. One of the things that we're currently looking at right now is looking at neural excitability within the cells. And specifically, we're trying to target more cellular processes like sodium channel proteins and how they can basically change, because there's some studies that came out and have been shown that the sodium channel proteins are these sodium channel proteins get expressed by virtue of like conditioning, depending on the type of conditioning, either fear potentiated style or Pavlovian fear conditioning paradigm. So we're basically maybe hypothesizing, trying to basically say that maybe the sodium channel proteins like this and sodium channel protein and a potassium protein might be involved in this rapid process that develops, but I'm really not sure because that's data that I'm actually like currently collecting in the lab.
Wonderful. If we have, we do have another question from Dory. If it's not the regular consolidation, where do you reckon the memories of the initial weak trial are stored?
That's a good question. When it comes to storage, one of the first things that I would try to think about is the hippocampus. And so actually, I actually just ran an experiment yesterday where I'm looking at the hippocampus, where I gave animals like the first training trial experience, which is kind of like the priming event, and took those brains and just like looking at downstream processes in some specific proteins in the hippocampus, like the sodium channel protein that I talked about. So probably maybe the hippocampus, but I can't say definite for now, but maybe when I get more results, then I will be able to implicate the hippocampus in this.
Wonderful. Thank you so much. And we do have not a question, but a comment from Braden that says, I wanted to just say you provided such great information. So that's lovely to hear. Thank you for that comment. Well, if there are no other questions, we are going to move on to our next talk. Thank you so much. Great. There we go. So our next series of talks will be flash talks, or data blitzes rather. And so each person will have 5 minutes and you will be receiving a message once you have 1 minute left. So thank you, Thomas, for turning on your camera, and feel free to start.
Thank you. So hello, everyone. My name is Thomas Burnett, and I'm a PhD candidate in neuroscience at Johns Hopkins University. My thesis project deals primarily with touch and how it informs behavior, but I have a special focus on ferrets and how they use their whiskers. And so I should preface that ferrets have been used for decades as good sensory and neurodevelopmental models, especially when it comes to vision research, but I feel like they offer a new avenue for understanding whisker somatosensation. Whiskers have been studied since at least 1940 in rats and mice for their notable barrel cortex and long whiskers, and they've become a staple of behavioral neuroscience. But there are certain questions that they can't answer. For one thing, how whiskers are processed in a non-barrel cortex, which are common among most non-rodent species. And so what I hope to find is that ferrets, ferrets do not whisk and they lack a barrel cortex. And so I'd like to see if I can use them to understand this sort of whisker somatosensation across greater mammals. And so beyond basic neuroanatomical work, almost no work has been done in whisker touch for ferrets. And so the method their brain uses is almost completely unknown. And so I'd like to discuss some of the foundational research that I've been doing this past year. We've largely relied on extracellular recording from S1, the primary somatosensory cortex, to understand how whisker responses are organized in cortex, and then how these neurons possess different response properties. And you can see the first results here. Next slide, please. Thank you. And so on the left, you'll see a ferret brain. In the inset are a series of dots. Each dot corresponds to a single neuron recording. Each color then corresponds to the receptive field in panels B and C. Blue corresponds to the body receptive fields, and the different reds correspond to the different facial receptive fields. And what I hope you can appreciate is that in the lateral S1, the representation is primarily for the face, whereas the medial S1 corresponds primarily to the body, similar to most other mammalian body maps. And one thing that's pretty notable, especially for the whisker somatosensory cortex, is that neurons appear to resolve in only multiple whisker receptive fields, as opposed to the single whisker receptive fields seen primarily in mice and rats. This is largely due to the fact that they lack a barrel cortex, which you can see on the right, on the rightmost part of the slide. On the top is a section of the whisker somatosensory cortex in ferret, stained with cytochrome oxidase, the classical stain for testing for barrel cortex. And on the bottom, you can see a panel from Lance and Simmons' paper showing the barrel cortex of a rat. And I hope you can appreciate the ferret cortex appears to lack the sort of distinctive cytoarchitectonic structure of the barrel cortex. And so we looked at other receptive fields that neurons appear to possess in facial somatosensory cortex. Next slide, please. And these were some of the results. Apologies for the loud colors. We tested a lot of neurons, and we were trying to make sure that we were able to convey all of them with different parts of the spectrum. But again, what I hope you can appreciate is that across the different whisker regions, each neuron appears to correspond to multiple whiskers, at minimum 2, often within the same row or within the same similar portion of the face. And so through these initial mapping studies, we've been able to understand how the neurons are organized in terms of receptive fields and which areas of the body they respond to. But one real question that we might have had, is that how are peripheral signals transformed to the central nervous system? Because whisker follicles contain dozens of different afferent types. These can be broadly divided into slow adapting receptors, which are responsive to continued force, and rapid adapting receptors, which are responsive to brief forces. And so we needed to parse this through a series of different whisker stimulations, which I can show you on the next slide, please. And here are some of the results of these experiments. These are a set of raster plots from different repeated whisker stimulations through a piezoelectric bender. From left to right, we have air puff deflection, a sawtooth deflection, where the whisker is slowly bent and then rapidly snapped back, and a sine wave deflection. And so in the cases of the air puff and the sawtooth deflection, we see robust responses to a quick, brief response of force. And so we believe this is indicative of a rapid adapting afferent being present in the cortical response. And on the right-hand side, we appear to have continued response to prolonged forces in the case of the sine wave. We also have similar results for a ramp and hold stimulus, where the whisker is deflected and then held in place and brought back. And we have those results forthcoming. Unfortunately, I couldn't fit them in this slide. And so we appear to have the different response properties and organization of S1, but we're still trying to understand how it's used in behavior. Next slide, please. And so for the future directions, we're hoping to understand how ferrets use their whiskers in behavior. I'm planning to use a prey hunting task for this. And we also plan to understand how different whiskers are integrated in cortex, especially in light of higher motion percepts like motion. And so final slide, please. Thanks, of course, to my mentors, Dr. Christina Nielsen and Dr. Jane O'Connor at the Mind Brain Institute. And if you have any questions, opportunities, or other collaborations you might want to start, you can reach me through any of these channels. Thank you.
Thank you so much, Thomas. We're going to move on to our next speaker, Amira Iman Hicks. So please take it away, Amira.
Hi, thank you. My name is Amira Iman Hicks, and I am a PhD student at McGill University. I'm studying, today I'll be presenting our project, Anatomical Organization of the Subfornical Organ, which is actually a project that we have in the final stages of preparation to be submitted and hopefully published. So next slide. So in terms of the subfornical organ, what is it? It is one of the brain's circumventricular organ, which share regions of the brain with an incomplete blood-brain barrier. But first, what is the blood-brain barrier? As shown here on the left-hand side of your screen, the blood-brain barrier is a protective mechanism that prevents the passive penetration of large molecules like hormones or pathogens from the peripheral circulation into the brain. It is made up of endothelial cells that are connected by tight junctions, and these in turn are surrounded by astrocytes. However, in regions of the brain called circumventricular organs, which are shown here on the right, here now the endothelial cells now have fenestra or pores that allow the passage of larger molecules, and they are also surrounded by a specialized type of glial cell called tanocytes. And another important cell type that's not shown here are pericytes, which are a type of smooth muscle cell. So next slide. In terms of giving a location of the SFO, this is a sagittal section of a rodent brain where the nose or rostral part is on the left-hand side of the screen and the caudal or tail is on the right-hand side of the screen. And essentially what you see is that the subfornical organ is a circumventricular organ that's found in the anterior wall of the third ventricle in the relative location of the hippocampus. And essentially it's considered to be a sensory CVO because it has a dense population of neurons where, in addition to having an incomplete blood-brain barrier, it's thought that these neurons can sense incoming signals from the periphery to then signal to downstream brain regions and regulate different physiological functions. So in our lab, the two functions that we are interested in is the role of SFO in regulating body fluid homeostasis because it has neurons that can respond to changes in osmolarity, as well as the role of SFO in regulating energy metabolism because it contains receptors for different energy metabolism hormones like leptin or ghrelin. And basically, while all labs have focused on understanding the diverse functions of SFO, no one has really answered the basic question of how is SFO organized? And that's what we decided to study or set out to answer in our study. Next slide, please. So one interesting piece of data that I'll be showing here is that we looked at characterizing the different types of vasculature or blood vessels in the SFO. So this is a coronal sections or 3 different coronal sections of the SFO where DAPI is labeled in blue. We also labeled for all blood vessels and all endothelial cells using an endothelial cell marker called RECA1, which is shown here in green. And what we found is that the SFO is highly vascularized with blood vessels, smaller blood vessels found in the center of SFO and larger blood vessels found in the periphery on the sides, which is outlined with that clear or unfilled white arrow. And interestingly, when we looked at fenestrated blood vessels using a marker called PV1, this labels the individual holes in those pores or fenestra. And what we found is that the fenestrated blood vessels are specifically appear in the medial and caudal region of the SFO. Next slide. And not only that, is that when we looked in further detail to characterize the different types of glial cells that surround these blood vessels, we found that when we labeled for a glial cell marker called GFAP, which is a classic marker for astrocytes, as well as vimentin, which is shown in the center column, which is a marker of tanocytes, we found that around the non-fenestrated blood vessels shown in the top row, these blood vessels are directly contacted by vimentin positive tanocytes, and these in turn are surrounded by GFAP positive astrocytes. However, when we compare this to the glial cells that are around fenestrated blood vessels, which is shown on the bottom row, here we see now that these glial cells express both vimentin and GFAP when we hypothesize that these are tanocytes, and that these directly contact the fenestrated blood vessels. So based on these results, we can conclude or we can hypothesize that the 2 types of blood vessels in the SFO express different types of markers and have different type of microenvironment that can regulate their function. Next slide. So this is just a summary slide showing all the different types of characteristics that we looked at. I'm not going to go through all of them, but essentially, if you want to know more, you can contact me or just wait for our paper to hopefully be published. Essentially, what we found is that there are differences in the permeability of the SFO as well as the vasculature. And we also found that the expression of parasites, that type of smooth muscle cell I mentioned earlier, were specifically found around the fenestrated blood vessels, but not around non-fenestrated blood vessels. And we also looked at the different types of neurons that are found in the SFO. Next slide. Thank you. And with that, just wanted to say that acknowledge my lab. Thank you so much to Masha, especially for allowing me to do this amazing project and get great images, as well as my undergraduate student, Simona Kobrinsky, who did a lot of the work in terms of neuronal studies. So with that, thank you.
Thank you so much. We are now going to move on to our next speaker, Tamara Parker. Take it away.
Good morning, everyone. My name is Tamara Parker. I'm a fourth-year PhD student in the Interdepartmental Neuroscience program at Yale University. And I'll be presenting my work entitled Right Inferior Frontal Cortex is Associated with Eye Movement-Related Brain Activity During Dynamic Social Interaction. Next slide, please. So under the direct mentorship of Dr. Joy Hirsch, I studied the neural correlates of live social interaction using functional near-infrared spectroscopy, or FNIRS. There are many advantages for using FNIRS. One advantage is that FNIRS allows us to analyze functional changes of social interaction in real time. Next, please. So specifically, it allows for regional and network analysis during live two-person studies. Next, please. So with this, you can be face-to-face with another person with optodes on your head, which measures hemodynamic signals or the bold signal in the brain. Next, please. With this, you can detect brain areas correlated with behavior, which is known as task-dependent activity. Next, please. And you can also identify what areas are dependent on other areas of the brain that are influenced by specific behaviors, functional connectivity. So some examples of task-dependent activity could be if you see activity in the right temporal parietal junction that's correlated with a behavior such as mentalizing tasks. And for functional connectivity, we can think of the right temporal parietal junction maybe working synchronously with frontal areas of the brain, such as the medial prefrontal cortex. Next, please. So our lab has been particularly interested in the mechanisms underlying live eye-to-eye contact. So what my particular question of interest is functional connectivity. I'm interested in what brain areas are connected during live eye-to-eye contact. Next slide, please. So in this particular study, we look at live person-to-person contact. So again, with FNIRS. And we have 46 participants participate in these different conditions. So they would either look at the partner's eyes, or they would look at the partner in a video, or they would look at a plant, because we want to look at a non-social condition as well. So next, please. So preliminary research in our lab has shown that eye-to-eye contact is correlated with hemodynamic responses in the right somatosensory association cortex, which is on the left, the supramarginal gyrus, which is also on the left, and then in the right, the dorsolateral prefrontal cortex. So again, this has been confirmed with eye contact, because we actually use eye tracking to really confirm that these areas are specific to eye-to-eye contact in the live condition. So what we're interested in is looking at these posterior areas. So if we go next slide, please. So we actually are focused on the parietal area, and we actually put 2 clusters in the parietal area. And we're interested in seeing how these posterior areas which are known to be used for sensory motor functioning, and how are they directly related to frontal lobe processes. So we want to see, are these areas connected? Do these areas work synchronously? So we see that using these 15 subjects that we see that these areas are working, but we don't know if they're working together. So this leads me to what my main question is, which is next, please. So I use PPI analysis, or psychophysiological interactions, to test functional connectivity in this parietal cluster using these 2 seeds. And specifically, I'm interested in the synchrony of these seeds from one area to the other. So our main question is, do these parietal areas work synchronously with frontal lobe processes? So next slide, please. So looking at the somatosensory association cortex and looking at the contrast eye to eye greater than others. So others is indicating eye to video as well as eye to plant, because we wanted to isolate what regions of the brain are interested in the realness of a face, because we don't want to just look at just faceness in general. And we also not really caring about the plants or the realness of a plant. We want to see what's specific to the realness of the face. So when we use the seed, we see, next please, that the somatosensory association cortex works synchronously with the dorsolateral prefrontal cortex and inferior frontal gyrus, showing reliable frontal parietal coupling. And similarly, next please, when we look at the supramarginal gyrus and place that as a seed using the same contrast, eye to eye greater than others, we see that a supramarginal gyrus works synchronously, next please, with the inferior frontal gyrus. So again, this is showing reliable frontal and parietal coupling in the brain. Next, please. So these findings support a network-based phenotype or functional connectivity in real life for real life eye contact and typically developing individuals rooted in the somatosensory association cortex in the right inferior frontal gyrus and the supramarginal gyrus in the right inferior frontal gyrus. So these results offer a novel neural circuits for studying atypical pattern of eye contact behavior seen in individuals with autism spectrum disorder. So this is ongoing work. So we're looking at individuals with autism spectrum disorder using these same three conditions and seeing, are we gonna see this coupling? So actually, if you look back a slide, we can see what we're really interested in seeing, are we going to see this frontal parietal coupling? And we hypothesize that we will not see this coupling between parietal and frontal areas and that there'll be an absence of this frontal coupling during this particular task. So we can go back again, next again. Okay, so this is just ongoing work that we're interested in. So then next, please. So yes, I would just like to thank my lab, the Brain Function Lab, particularly my advisor, Joy Hirsch, Shen, who's helped me a lot, and Jen, as well as other members of the lab. I want to thank NSF-GRFP, my funding source, IMP, and if you are interested in anything that I've talked about in our lab, you can use the QR code that's in the right-hand side, or you can email me at tamara.parker@yale.edu. Thank you for your attention, and I'm interested to hear any of your questions.
Great, thank you so much for all of our speakers. We are now going to enter into a Q&A for the talks that we have just. So I will ask the questions that have been asked by our audience on screen to answer them. From Dory, they ask, did you in this brain area? So
Hi, sorry, you were cutting off before, so I wasn't sure if you asked the question.
Yes, did you see the question?
Yes, I see it. So any influence of presence of parasites on the glioma? So for parasites, what we used as our marker was NG2, and NG2 is expressed by both parasites as well as NG2 glia. So right now we only characterize how they, like where they are around these fenestrated blood vessels, and we didn't yet see how they can influence. So that's the next upcoming question, is how can NG2 glia or our parasites affect the vasculature of the SFO?
Great, thank you. And please let me know if I cut out. I will re-ask the question. Thomas, you did have a question which you answered in text, but I think it would be great for the audience to hear. So I'm just going to re-ask it. So Julie asks, ferrets seem like a great system to study whisker sensory input. Do you think it is a better model to study this versus mice? What questions do you think are best to study in ferret versus mice?
Yes, thank you. That's a great question. One of the major pluses of ferrets is the fact that they are neurodevelopmental models, like I previously said. And another is that they're actually neuroinfectious models as well. They can catch many of the viruses that we do, such as tuberculosis and notably SARS-CoV. So some questions I believe will be better in ferrets, such as how gyrification of the cortex will affect sensory development processing. But for my research in particular, I believe that the fact that ferrets don't whisk and thus lack some of the proprioceptive contamination that sometimes happens with sensory signal, I believe that'll lend them as better models for certain questions concerning especially integration of touch signals. So motion, which requires the integration across time and space, is one of the questions that I hope to answer in ferrets rather than mice.
Great, thank you so much, Thomas. So our next two questions are for Tamara. from Jonathan, who asks, what is the resolution compared to fMRI? Can it be mapped to patient's own MRI?
Oh, that's a very great question. So yes, with FNIRS, unfortunately, we don't have the same resolution capabilities of fMRI. However, we do use a technology for us to map the brain. So after the person does interact with another person in the social interaction experiment, we also do this cool thing where we use an iPad and we try to map kind of locally where the brain is so that we can know exactly where the channels are. But unfortunately, we don't have the same resolution as fMRI, but we do have the advantage of at least, having two-person interactions. You can actually see a person, which you cannot see in fMRI because you'll be in the scanner.
Okay, great. And Jonathan has another question for you. How do you suppress cortex controlling eye movement, which can be active even without eye movement?
Sure. Yes. So I guess the way our task design is, it allows us to really understand, okay, so the way we do this is we have these 15 seconds, but what happens is that there's 3 6-second cycles in which the gaze alternates either at the eye or either alternates at the a light of the subject. So we wanted to really use time locked capabilities. And we also used eye movement. We also use eye tracking as well to confirm it. So we not only were just looking at, okay, this is when they're doing, this is when they're supposed to do the eye task, but we were interested in seeing, okay, when we see that they look exactly at the eye box, like what exactly is going on in the brain. So we're really interested in this specific mechanism.
Great, thank you so much. Amira, we have another question for you. Julie, who says, great talk, love those CNS blood vessels. Is there evidence that tanocytes can regulate vascular features like blood-brain barrier leakiness or other properties that make sure that neurons in the SFO can sample the periphery?
Yeah, great question, and thank you. That's actually one of the main things that the lab of Vincent Prévost in France, he studies how tanocytes can regulate the blood-brain barrier features of the median eminence. So the median eminence is another circumventricular organ that's essentially connected to the pituitary gland. So that's a big thing that his lab studies, is trying to see how tanocytes can regulate blood-brain barrier features. And one major thing that he found is that tanocytes can regulate how hormones are secreted out. So that's one thing to mention, is that the median eminence is a secretory CVO. So it's not where things are uptaken, but where things are taken out, or secreted out of the brain. So essentially he studied that. And one feature, for example, is he's shown that tanocytes can regulate how gonadotropin releasing hormone, or GnRH, is released from the brain, depending on the estrous cycle or the reproductive cycle of mice. But essentially, this question of how can tanocytes regulate uptake of signals is what I'm trying to answer in my PhD. So I will let you know soon, hopefully.
Great, we look forward to it. And the last question will go to Tamar. Well, there's actually 2 questions, so we'll ask the 2. To those who are keeping time behind the scenes, let me know, because we do have time for those. But one of the questions, Tamara, Nikki says, thanks for the interesting talk. Sorry if I missed this. I was wondering how you selected your ROIs. Did you use data or hypothesis-driven approach?
Yes. So actually, we did a data-driven approach because we have another study that's ongoing where they're looking at typically developing people, so about 15, and ASD subjects. And what we saw in that preliminary data is that we saw that people who so basically what happened is that the people with autism had an autism diagnostic observation schedule, which is trying to see how autistic they are. And we saw that the ADOS correlated with these areas, the somatosensory association cortex and the supramarginal gyrus. And we saw that, okay, that's interesting. It seems like when people who are probably less social or have autism seem to have like a suppression of these areas. And this only was seen when we had eye tracking that was confirmed. When they're looking at the eye box, it seems like these areas of the brain seem to be really active for typically developing individuals, but in autism, it's not. So we want to test further the question of seeing how does these areas, is this like a a region-specific problem, or is this a network problem that we're going to see in autism? So I think.
Great, thank you so much. And so we are running out of time. 2 questions. And if you can type them and respond to them, that would be really, really appreciated.
Yes, I will.
So thank you to everyone who was a speaker. That was a fantastic session. So virtual round of applause. We are going to be moving on to our next series of talks. And next up, we have a Sequoia Bell who has videotaped her talk. So I'm going to get to that.
Hello, my name is Sequoia Bell, and I'm a Laney Graduate School Global Scholar, and I had the opportunity to work with Dr. Malavika Marubian, who is an assistant professor in the biology department at Emory University. During the summer, I was able to work with the Laney Graduate School on during the research program at Emory University. The name of my presentation is The Neural Correlates of Social Interaction, and in this lab, our goal is to connect neural activity to social interactions. So first, I would like to present some background information on the project that I was working on in the summer. So engaging in social interactions are one of the key motivators in our everyday life. Human beings have social interactions every day, like speaking in front of an audience, talking to your Uber driver, or a mother holding her baby. But basically, social interactions play a huge part in our lives. So just like humans, animals also have social interactions with one another, and it's crucial for the survival in most mammalian species. Despite the importance of social interactions, we know little about how the brain drives these complex behaviors. This is something that the lab hopes to address. So in the lab, the mouse is used as a model system to study social interactions. My project was then to quantify these social interactions that the mouse engages in to later relate to neural activity. So why use the mouse as model systems? So we wanted to use mice because mice are involved in positive social interactions such as sniffing and grooming. And so it is reward, and dopamine is released in the nucleus accumbens, which is the reward circuitry, and social interactions are often complex and highly dimensional. So relating activity in the brain to specific behavioral actions can be challenging. Therefore, finding a way to quantify behavioral actions would be necessary to relate them to activity in the brain. So social interactions have such a wide range and are not so easily pinpointed. So therefore, we quantify specific social interactions in mice to begin with correlating them to neural activity. But the first step was to identify, to analyze, and to code these behaviors. So in our first step, we did a frame-by-frame annotation of the mouse behaviors in a home cage assay. So in the home cage assay, the mouse of interest, which is the test mouse, is allowed to freely interact with a juvenile mouse, which is the target mouse, introduced in his home cage. We classify the behavior of the test mouse into various categories. So this video that I'm showing you shows the social interactions between 2 male mice. As you can see, they are engaging in behaviors such as sniffing, grooming, and pursuing. And this is when one mouse comes directly in contact with the other mouse to engage in social interaction. And so the application that we use is the Boris application. And when we target a specific mouse, we use this application to analyze its behaviors. We use a specific name and a specific code. And so then we use the amount of time spent in doing this specific behaviors recorded, and how many times the behavior occurred is also recorded. And now I'm just going to briefly break down the behaviors that we use and the difference between social interaction behaviors and solitary behaviors. So basically, behaviors in which the animal is actively engaging in interactions with the other mouse are called social interaction behaviors. And then solitary behaviors is when the mouse carries on its own without engaging with the other mouse. So the social interaction behaviors are pursuit, anogenital sniffing, face sniffing, other sniffing, and grooming. Pursuit is when the behavior starts when the mouse approaches and ends when he makes contact with the other mouse. Anogenic sniffing is when the mouse is sniffing the tail or anus area of the other mouse. Face sniffing is when he's sniffing the face or facial area of the other mouse. Other sniffing is when he's sniffing the mouse anywhere else. And then grooming is when the mouse places his paws or snout on top of the other mouse's body. And then example of solitary behaviors are just walking, rearing is when he's on his hind legs or feet are out, and then sitting. And so now I'm going to briefly go over the actual data that I got from watching and analyzing these mice and their behaviors. So as you can see, I've broken down every single behavior that I use and then did the percentage of each mouse of how long that mouse spent doing that specific behavior. And so basically the purpose of us doing this was to demonstrate how heterogeneity in mice engaging in different types of behaviors. And so basically it's helping us having a nuanced understanding of behavior and will help us to relate this behavior to neural activity, right? And so this was our overall goal. And so through these trials, you can tell the differences in social interactions and needs for interactions in the mice that were specifically targeted. And lastly, our next step is the last step that we're going to do after we've taken these behaviors and quantified them is like in the future, we hope to compare these manually annotated behavior measures to results from novel, fully automated behavioral annotation methods to determine the usefulness and accuracy of these new methods. And so basically, overall, we having a detailed understanding of behavior, mice engaging, will help better relate activity in various parts of the brain to specific aspects of behavior and understanding how the brain drives these complex social interactions. We want to compare the methods that we use manually to the methods that we use that are fully automated, such as recording the neural activity through the helmet that we put on the target mouse to track its social behaviors and neural activity. Then we can use these methods that were fully automated to relate activity in various aspects of the brain. And that is just basically our overall goal. Is like, what does the brain do that drives these specific behaviors that help us engage in social interactions with one another? And then understanding, you know, overall just how the brain works in driving these social interactions. So yes, and that would be the end of my presentation. Thank you, Black and Neural, for inviting me and letting me to share with you this research that I've done. It was a complete pleasure. I would also like to give a thank you to Dr. Malavika Murugan from the Laney Graduate School Summer Program that I was in, and Dr. Amanda Marie James, who was also over that program. Thank you very much.
All right, well, thank you. We are now going to ask our next speaker, Tia Riggins, to come up and give your talk. And I apologize, Tier did record her video, but we'll be here for the Q&A. Sorry, Tier.
Hello, everyone. My name is Tia Riggins, and I'm a PhD candidate in the Regenerative Electrode Interface Lab at Michigan State University under the direction of Dr. Erin K. Purcell. And I'm going to talk about my talk, which is Neurotechnologies Design Features Impacts on the Identity and Function of Reactive Astrocytes. Imagine suffering from a stroke and not being able to move your arms, legs, hands, or feet for over a decade. Imagine finally regaining some ability to do a simple task all over again after major brain surgery and a month of intense therapy. Judging by the look on this woman's face, implantable probe research has the ability to change lives. However, there are concerns to improve the stability of chronic recordings. One way is to investigate biological response as an avenue to improve chronic signal-to-noise ratio. There's an ongoing question between response and reporting quality observations that varies across the lab. But in this paper, there demonstrates a clear relationship to biology being a contributing factor. In this study, 16-channel single-shank silicon-based microelectroarrays were explanted from 4-week-old rats. And they were able to determine that not only were microglia and gliosis fragments on the explanted shanks, but also in the area where the implants were indicated in orange, astrocyte reactivity was the most dense and accompanied with neuronal loss. In a separate study, ND stands for neuronal density and NND stands for non-neuronal density. The green stained cells are neurons and the red stained cells are astrocytes. And these are endpoint histology that demonstrates that recording quality decreases with a higher non-neuronal density. So why do reactive astrocytes appear so numerous around the wound site? Well, normal astrocytes are responsible for brain homeostasis and immune response. They communicate back and forth with neurons, and they adjust the brain microenvironment to suit the neuron's needs. They do this through sensing biochemical and mechanical changes. And if there's insult, specialized proteins called G-coupled proteins at the ends of astrocytes become phosphorylated. So based on the injury, reactive sites are created. And if it's something like mechanical damage, nuclear factor kappa beta becomes phosphorylated, which binds to DNA and starts a cascade to release neurotoxins. A way that researchers have been trying to get around this issue is through the production of next generation probes. So in our lab, in collaboration with Winley and the Fraunhofer Institute, we've micro-machined diamond probes because diamond material provides material advantages such as good biocompatibility, wide potential window, and low double layers capacitance, amongst other attractive features. And because of its microfiber size, the probe is also able to sense electrochemical changes on a physiological relevant level. And it can also detect single cellular concentration of dopamine. Using new probe materials is a way to investigate how we can better integrate tissue function at the probe interface. My project specifically ties into using this diamond probe as well as other commonly used materials. Our lab has demonstrated that local shifts in ion channel function accompany implants, and I'm investigating or expanding upon this work by pursuing the hypothesis that changes in astrocytic ion channel expression accompany reactivity surrounding these electrodes. During the remainder of my PhD and the F99 phase of my funding, I've tested this hypothesis by using immunomethods to develop an inflammatory model that changes morphology based on stimulus and injury. I've also used patch clamp techniques to determine the firing health of these models. And I'm also using PCR methods to verify that these models have upregulated genes that are specific biomarkers to this type of reactive astrocytes. Here, I verified that specific gene called complement component 3 is specific only to neurodegenerative reactive astrocytes, as opposed to any other astrocyte type. I'm also using atomic force microscopy to characterize astrocyte interaction with probe features and to verify that astrocytic stiffness increases with probe bending stiffness, which changes its morphology. In addition, I'm also investigating piezoelectric force microscopy to detect signal propagation of astrocytes. In summary, device features and bending modulus of materials have influence on the expression and reactivity of astrocytic activation. I'm thankful for this fellowship, and I want to build upon this work in my K0 phase during my postdoc of the following 4 years of funding. Thank you.
So thank you to Thierry. I would also like to say that for our session 3B, we do have 3 new judges. So we have Jessica Falcone, who received her PhD from Georgia Tech in neural engineering and is currently a technical sales engineer at TDT. We have Dr. Conrad Cording, who is using data science to understand brains with a recent focus on causality, and who is also the lead of NeuroMatch. I'm sure many of you know. And we also have Dr. Danielle Gonzalez, who is an HHMI postdoctoral fellow at Purdue University and specializes in developing neurotechnology and neural interfaces. So we're going to go on to our next talk, which is also pre-recorded. We have Oluwasi Bun Timothy Akinii. And
good day to you all.
There we go.
I'm presenting the development of a robotic neurorehabilitation device for stroke survivors in a resource-poor environment. I am Oluwasegun Akinii. Stroke, a neurological dysfunction, is a global burden. In fact, Sub-Saharan Africa has the highest number of stroke occurrences and mortality in the world. In most cases, stroke leads to loss of motor function, subsequently affecting the activities of daily living in stroke survivors. Studies have shown that conventional neurophysiotherapy aids neuroplasticity in stroke survivors when they undergo the process of relearning new tasks. Unfortunately, regions with the lowest physiotherapist to patient ratio currently have the highest number of stroke-related deaths worldwide. For instance, Nigeria has less than a physiotherapist to 10,000 patient ratio, which is one of the lowest in the world. Rehabilitation robotics is a developing field with numerous advantages. Although there is this big question of the superiority of robotic therapy to conventional physiotherapy. To us here in Africa, we hold on to the narrative that rehabilitation robots can help us address the issue of shortage of physiotherapists on the continent. Pulsar is a hand rehabilitation robot for passive and active rehabilitation sessions, as you can see in the video here. During sessions, the muscle that controls the upper limb, that's the hand, is being stimulated by a functional electrical stimulation device. In addition, we are incorporating EEG, electroencephalography, to detect patients' intent, especially in patients who have flaccid limbs. I showed the video of Pulsar version 2 in the previous slide. Currently, we are undergoing usability tests, comparing Pulsar version 2 that we recently completed to Pulsar version 1. And we had 5 subjects who went through some preliminary tests. We did satisfaction tests, appearance tests, and performance tests in order to see how satisfied patients are using the robot, the appearance of the robot to them as compared to the previous version, and the performance of the robot in moving their arm, executing basic tasks. And we're able to get a result as displayed in the chart here. After this study, we'll be going to several other tests and we are hoping to commence clinical trials in about 3 months from now. Thank you. Thanks for your attention.
Okay, up next we have another pre-recorded talk by Wisdom Ikezogwo.
Hi, and thank you for having me. My name is Wisdom. I'm from the BC Lab at Obafemi Awolowo University. And today I'll be giving a presentation on how we supervise domain generalization for integration of disparate scalp EEG data sets for epileptic seizure detection. So EEGs allow us to acquire brain activity data. And epilepsy, a neurological disorder, shows up on EEGs as periods of abnormal brain activity, causing seizures and having the outward effect of sporadic body movements and uncontrolled body movements actually, and memory loss, subtle memory loss. So a cool thing to do would be to be able to detect or predict the onset of epilepsy, of these seizures. And so in literature, what you find is different studies that do this. They curate their own data sets and go on to develop models and classifiers that either predict or detect or do both the onset of epilepsy. Sadly, the models generated or developed do not generalize well, do not perform well on other data sets. And this has been identified to be caused by the distributional shifts between these data sets. So down below, we can see the CHP MIT data set on the left and Temple University's TUSZ on the right. And while the latter is a Gaussian distribution, the former is a double exponential one. And so 2 reasons have been identified for this. First being that out of the underlying brain aerodynamic feature space, the subsets that each data set represents is different from the next. And so what this means is that they are gotten from different patients, different subjects. And the second is that the acquisition and processing pipelines are quite different. So they're different in the number of electrodes that they use, placement of those electrodes on the head, sampling frequencies, and a host of other differences. And so a common theme to solving this problem is to create even larger and larger data sets. And a lot of studies do this via data augmentation methods, or just brute force curating even increasingly larger and larger data sets over time, like Temple's TUSZ. While these are great, they don't quite solve the problem of integrating multiple disparately curated data sets in training a single generalizable model, and in turn increase the clinical deployment, adoption, and use of these models. And so we try to solve this, framing it as a supervised domain generalization problem. So if we look at each data set as belonging to a particular domain and label all but one, the source domain, and label that one, the target domain, the SDD task is then to learn a labeling function that is generalizable to the target domain, but trained on the source, on samples from the source domain. And so we achieve this by first converting converting the EEG time series data to images, and we do this in a way that preserves both the spatial and temporal information. Next, we use the transfer component analysis based dimensionality reduction technique to extract 10 components from our data. And these 10 components then go on to create a 10-layered image. So you can view this as the 3-layer RGB images we commonly find in the world. And this then go on to train the recurrent convolutional neural network. And this particular network was chosen specifically because it allows us to learn from both the spatial and temporal information within our data. So our results gotten from privately collected data sets with our collaborators at the University Teaching Hospital called OAUSF1 show that this is indeed a viable approach as it compares well with previous large-scale validation studies. And while Gabor and Hofstengardner's study performed significantly better than ours, they reduce the shift between the training and validation sets ab initio. So what this means is that they either use the same acquisition pipeline or the same processing step afterwards for both data sets. And so Reveal is a much better benchmark. Conclusion, although much work is needed to improve the technique, the results do show that reduction in the domain shift between data sets can indeed lead to an increased training data and consequently increased model generalization. I'd like to thank Dr. Kayode Ayodele at the PC Lab, my PI. I'd like to thank Professor Komolafe from the university's Faculty of Clinical Sciences, and I'd also like to thank Professor Nkumbono from the University of Wollongong School of Computing and Information Technology. Thank you very much.
Okay, well, thank you. Our next speaker is Sylvia Edo-Igi Awari. And I tried to pronounce it as it was phonetically sounded.
No problem. Yeah, I don't know if I can, should I also unmute my video, I think, or?
Yeah, you pull up your camera.
I've tried, but it's saying I can't do it. But,
Here, give us a second.
All right, thank you so much. Okay, hey guys, thank you for listening to my talk. I'm gonna be talking to you about comparing the amplitude. Is there a way that I can also say next slide? There, thank you so much.
Yeah,
I'll say next slide for each.
Yeah, there you go, just say next slide.
All right, thank you. So I will be talking to you guys a bit about my project, which is comparing the amplitude integrated EEG, or AEG as I'll be referring to it for this talk, to another algorithm called the Tiger-Huang algorithm for neonatal seizure detection. So that's quite a mouthful, so next slide. Next slide, please. So that's quite a mouthful, so I'll give you guys a little bit of background so you guys can understand exactly what I am talking about. Next slide. So first of all, neonatal seizures are actually a huge problem. And that is because seizures have the highest occurrence within the first month of life. And that's often because of complications that arise during labor and delivery. Next. And so as a response to these complications, clinicians use something called 24-hour EEG monitoring. And it's basically to monitor the brain development to assess for how the baby is actually doing over time in order to prevent devastating neurological outcomes. And so the problem with 24-hour EEG monitoring is that it generates huge amounts of data. And on average, around 57,000 pages that actually has to be manually observed by clinicians. And so there's a need for sensitive and specific algorithms that are actually able to assess EEG real time. Next, please. So the current faulty solution that we are using is something called amplitude integrated EEG, or AEG. And it's faulty because it has low specificity, has a low kappa score, which is a metric for inter-rater agreement. And it also only looks at amplitude trends. Next. So I propose to compare AEG to something called the Tiger-Huang transform, which is more optimized for analysis of nonlinear and non-stationary signals like EEG. Next. Next, please. Thank you. So next, I'm going to walk you guys just briefly through what we're actually doing when we compute the AEG. so you guys can get a sense of what it's good for. And so next. Okay, so basically what AEG is, it's a series of transformations that have been applied to the raw EEG. So as you can see here, at the very first panel is just the raw EEG. I've marked where the onset of the seizure is. And so we apply these transformations, and the goal really is to get a sense of the amplitude trends in the EEG that clinicians can now use as a proxy for seizure occurrence. So next. So what is most important to really take away is that clinicians are trained to look at a rise in the baseline amplitude of the lower terminal. And that is basically used as a possible indicator of seizure occurrence. And so here I'm actually looking at 15-second windows. So each line that you see in the terminal points at the very bottom graph, they're called terminal points. And basically it's showing me the span of amplitudes across 15-second windows. Generally, we use 5-minute windows, but I've actually shortened the resolution because I want to see how can AEG perform at its very, very best. Next. And so what I've used is this as my feature. Next, please. Yeah. And so now I'm going to just walk you guys briefly through what the Tiger-Huang transform does. Next, please. So what it can do is it can take a multi-component signal and break it down into its individual components. And then we can, next please. We can actually compute the energy associated with each of those components and then take windowed portions and compute the mean log of that energy value and compute something called the ANE, which is the average nonlinear energy. Next, please. And so that is going to be the selected feature that I'll use for my random forest classifier to compare AEG to this Tiger energy. Next slide. Yeah. So just as a reminder, for the AEG, I've looked at the lower terminal as my feature that I'm going to be using to assess seizure classification. Next. And so what my data set was quite small. So we're working with 4 seizures, 1 neonatal patient, and a 42-minute recording. Next. But we see that AEG did perform not bad. Next. The F1 score was pretty good. And next. The accuracy was pretty good. But I want to just remind you guys that I'm actually like using an AEG with a much higher resolution than would be to actually use clinically. So this is kind of like AEG performing at its very best, right? But next slide. Even at its very best, it still underperforms in comparison to the Tiger-Huang algorithm. And so as you can see, the AUC is much higher, the F1 score is much higher, and the accuracy is much higher. So this leads me to the end of my presentation. And our future directions are to, of course, increase our sample size and also compute more features that might be more robust. And that's all. Thank you so much for listening.
Thank you so much for that talk, Sylvia. So we now have our Q&A sessions for our 4 speakers. We are going to cut the Q&A a little bit short because we are running a little over time. But for those who do have a question that isn't asked, we would appreciate if you can type in your answer. Thierry has already done that. So for those who are interested in her question and response, please go ahead and look at that. We have one for wisdom. Conrad asks, I'm curious how big the amounts of domain shift are across labs. Any chance you could estimate? So I'm going to ask Wisdom if you are available to answer that question. Please come up. Thank you, Wisdom.
Hi, can you guys hear me?
Yes.
Okay. Thank you for that great question. So we used a 2-sample test. To basically check between the TUSZ and CHPMIT. But we haven't done like a test amongst all of the different data sets. So from the bond data set all the way to Temple's data set. But like from my presentation, I showed just the FP1 and F7 electrode pair data, and I showed like the distribution shift between those two. In the actual paper, we tried to resolve this using like an averaging method. So I think I'll put a better answer in the I'll type the better answer down below. Thank you.
Great, thank you. And Sylvia, we have one for you. Conrad asks, I'm curious how much extra mileage there would be using a broad feature set and doing advanced machine learning. Also curious about the cross-validation strategy.
Me unmute myself. I'm actually really new to machine learning, so I would definitely be interested in your ideas concerning the cross-validation strategy. I think there would definitely be more mileage, and we could definitely create a more robust algorithm by increasing the number of features that we have. These results are very preliminary, but they're good. So I'm really confident about what we can do for better detection.
Great, thank you so much. Wisdom, we do have a few more questions for you, but I'm going to ask that you type in your response so we can move forward with the next set of speakers. But thank you to everyone who asked your questions, and we will make sure that they are responded to shortly. All right. So next up, we have Tyler Bonin, who has a pre-recorded talk for us. Okay, Tyler, it seems like something is a little off with the video right now. We're going to come back to you, and we're just going to move to our next speaker so we can figure out what is going on technically here. So our next speaker is Armel Nisiangani.
I'm actually impressed you attempted my last name. Can you hear me?
We can, but there's a bit of buzzing in the background.
Sorry, there is the guy slightly better. I'm gonna move my microphone right next to my mouth. This way you guys can hear me better. Better?
I'm sorry, you're still cutting off.
No.
Oh, do you want to try speaking then?
Wait, am I still cutting off, or are you still hearing like noise in the background?
The noise is now gone. You sound great. So I'll let you know if there are any other difficulties. Thank you, Armel.
Okay. All right. Sorry about that. If anything, I want to send someone real quick down there to like ask him to stop. All right. Good. All right. Hi, everybody. So this is Armel, and today I'm going to present some data from my graduate project, which is titled Systems and Methods to Quantify and Improve the Intrinsic Signal Imaging of the Mouse Visual Cortex. All right. Next slide, please. Next slide, please. All right. So let's start with the definitions. What is an intrinsic signal optical imaging, or ISI in short? It is a method that measures blood responses resulting from a neuronal activity. In other words, it is an indirect measurement of neural activity. Here, what we do is that we investigate the process's vision. This area is also known as the visual cortex. Each section of this area responds to specific spatial aspect of vision. eye size allows us to determine which section of the cortex respond to specific spatial locations. This is what we call mapping, and the resulting are retinotopic maps. Our experiments were performed on mice, which have become an important tool for studies of normal brain functions, as well as neurological disorders. Our use of eye size is shown right here on this panel, on the front panel. We are actually able to place a camera on top of a cranial window, which is shown right there, which give us access to the brain and allow us to record blood responses. During a typical experiment, a visual stimulus moves either horizontally, left to right. Oh no, I'm still in the first slide. Sorry. I'm still in the first slide. There we go. All right, so during a typical experiment, it will move or either horizontally or vertically. And the results or maps are actually shown right there on the right. Neural activity can then be inferred from those blood responses. However, these responses are often noisy. This is because blood is affected by many other physiological phenomena, such as heart rate, for example. So what we try to here is answers two questions. First is how much data is needed for a reliable map. Second is how well does eyesight reflect neuronal signals. Next slide, please. All right, so let's answer the first question. How much data is actually needed? For the sake of here, we have defined one sample as one visual stimulus sweep, and each sample actually lasts for 18 seconds. 180 seconds was a typo. Area centered from a reference map is that the error reached 100 micrometers and below when at least 50 to 60 samples were averaged to create a map of the primary visual areas. But for higher visual areas, we've actually noticed that we need, and the creation of a robust map necessitates somewhere between 70 to 80. For the second question. All right, so for the second question, we try to determine how well does the eyesight reflect neuronal signals. As shown on the left panels, we were able to actually perform craniotomies right after the maps were generated. The location of the craniotomies were then aligned, which is shown on the bottom part of the left side. They were aligned in order to generate the coordinate from the eyesight and neuronal responses, which allow us to generate a coordinate for electrophysiology. From there, we're able to plot the correlation map, which is shown on the right-hand side. And these correlations show a high correlation between the coordinates of the intrinsic signal, as well as the electrophysiology. I do have to note that this result was found in the superficial cortical layers, but we did not find the same result in the deeper layer. Next slide. All right, so in conclusion, we found that 50 to 60 samples are actually required to generate a reliable map of the primary visual areas, where 70 to 80 samples were enough to create a repeatable map of the higher visual areas. We also found a high correlation between the electrophysiology and eyesight coordinate, but this was specifically found in the superficial cortical layers, but not in the deeper cortical layers. So in conclusion, our method, along with all these results, will help us to further extend our understanding of the representation of neural activity by intrinsic signals. Next slide. That's it. All right. Thank you. I would like to thank the Hydros Lab at Georgia Tech, as well as the Black in Neuro organization for this opportunity. Thank you, guys.
Okay, thank you so much. We are going to try to get Tyler's presentation up. So give me a second here to just switch screens. And Tyler, if you'd like to turn on your video right now. Oh, sorry, you know what, it's fine, we're good. Okay, let's see if this.
He, him. I'm a graduate student at Stanford, co-advised by Anthony Wagner and Dan Yemens. Really looking forward to sharing some of our recent work and grateful to be in community with each of you. The medial temporal lobe is a network of neuroanatomical structures often implicated in spatial and memory-related behaviors. However, there's an enduring debate surrounding their involvement in perception. For over 30 years, this debate has centered on perirhinal cortex, a medial temporal lobe structure that receives inputs from many high-level sensory systems. Standard view is that perirhinal cortex is involved in memory-related behaviors, not perception, while an alternative hypothesis is that in addition to its mnemonic functions, perirhinal cortex supports so-called complex perceptual representations. Remarkably, and unfortunately, decades of lesion studies have been used to both support and refute the involvement of perirhinal cortex in perception. Today, I'll suggest that these apparent inconsistencies can be resolved by situating experimental behavior directly in relationship to the performance supported by canonical sensory systems. And I'll use this slide to sketch that idea out visually. We can think of performance supported by the visual system on the x-axis here, the ventral visual stream or VVS, going just from chance to ceiling performance. And on the y-axis, human performance, again going from chance to ceiling. If behavior fell along the diagonal, it would suggest that our performance is supported from a linear readout of the ventral visual stream. However, the hypothesis in question suggests that perirhinal cortex enables neurotypical subjects to outperform the ventral visual stream, while perirhinal lesion subjects revert to it. Critically, if the ventral visual stream supported performance as at ceiling, then these tasks are not relevant because no additional perceptual processing would be required. Now, that's just the idea that we're using to hopefully resolve this debate. Methodologically, the move that we're making is that we're going to be using task-optimized convolutional neural networks as a proxy for the visual system. This will enable us to predict ventral visual system supported performance directly from experimental stimuli. And we'll call that model performance. To compare model performance with perirhinal lesion and intact behavior, we're going to leverage this approach first retroactively, collecting stimuli and experiments from experiments that have previously been published, right? These are experiments that have been used to both support and refute the involvement of perirhinal cortex in perception. We're going to estimate model performance in these experiments directly from their stimuli on a trial-by-trial basis using the following protocol. Given a stimulus screen, for example, this one, where subjects are directed to pick the odd one out, we're going to segment this trial into n independent images, and we're going to present these images to a computational proxy for the ventral visual stream. Happy to talk about this in detail in Q&A. We're going to extract n feature vectors from this model, compute the pairwise correlation coefficient between these vectors, and identify the item with the lowest off-diagonal correlation as the oddity, and mark this trial as either correct or incorrect, depending on its correspondence with ground truth. Now, we can do this not just for a single trial, but for a whole experiment for each of the experiments that we have across all of these studies. And then we can compare model performance with perirhinal lesion and intact behavior. What we find is that perirhinal lesion subject behavior is very well predicted by this computational proxy for the ventral visual stream, while perirhinal intact behavior diverges both from the model and perirhinal lesion subject behavior. And critically here, we also observe that there are multiple experiments that have been misclassified on both sides of the debate. That is, these are experiments that while they might be described as complex, they in fact are not useful for us here. Now, there's a bunch of limitations with this retrospective analysis, so we decided to collect a novel data set that allowed us to compare human behavior with electrophysiological recordings from high-level visual cortex, that is IT or inferior temporal cortex. What we found was that high-level visual cortex is well approximated by our computational approach, not surprising for us. Moreover, perirhinal intact subjects are outperforming here direct electrophysiological recordings from IT. right? This is a direct comparison of our main hypothesis. Moreover, divergence between IT-supported performance and IT scales with reaction time. Largely, these results validate the previous retrospective analyses. So in summary, our computational approach enabled us to resolve decades of apparent inconsistencies. That is, a computational proxy for the visual stream predicts perironal lesion subject behavior, while perironal intact performance outperformed the ventral visual stream. And to summarize this, we could say that neurotypical visual discrimination behaviors depend on perirhinal cortex. This work highlights the utility of using biologically plausible computational approaches that characterize neural function directly from experimental stimuli. Thank you for your attention and their time. Thank you to the labs that I work with, the original authors, my funding sources, and I'm really looking forward to your thoughts and ideas. Thanks so much.
Okay, thank you very much, Tyler. Next up, we have Harper Weigel.
Hi, can you hear me all right?
Yeah, you sound great.
Cool. Okay. Next slide, I suppose. I'm Harper. I'm a undergrad at Carnegie Mellon University, and with Lily Widmer, Taylor Abel, Sean Kelly, Polka Grover, Ashwati Krishnan at the Neural Web Research Lab at Carnegie Mellon. We've been developing a new EEG device that can be used for long-term recordings, rapid application, and simultaneous recording with scalp obstructions. Next slide, please. A little bit of background about EEG. It is a non-invasive neural monitoring device that is used by applying electrodes to the scalp and in a uniform spatial sampling distribution. This is called the 1020 system. That is depicted in the top right picture on the slide. In the bottom right, we have a 1010 system, which is a higher density version of EEG. In the top left picture, you see a conventional EKG electrode being used on the scalp to showcase what a scalp obstruction might look like. And then EEG is able to monitor the scalp by cross-referencing electrodes across hemispheres. This allows you to determine any anomalies in the brain, such as concussions, tumors, lesions, or anything that would cause the brain to produce an asymmetrical signal, but you can also do a frequency analysis of individual electrodes, and from that you can determine different components of the brain, such as alpha waves, which are typically associated with concentration. Next slide, please. One of the primary issues that technicians have found in applying modern EEG is it can't be applied if there are scalp obstructions because it's often a cap that is pulled across the whole head. So if a patient is using stereo EEG or another obstructing or invasive neural monitoring device, and they can't take advantage of EEG, which could be used in tandem. Also, large hair, such as African American hair, is typically not conducive to using EEG, and patients with large and obstructing hair have to be turned away from EEG patients, creating an inherent bias in the neural monitoring community. Another issue that is commonly found is long-term recordings with EEG require the EEG to be removed and rehydrated if they're using wet electrodes and then reapplied to the scalp afterwards. Application of the EEG can take from 20 minutes to an hour. And so having to do that intermittently throughout a long-term test creates discontinuities in the signals and makes the data less useful. An alternative to this is dry electrodes being used in the scalp. However, dry electrodes have a higher impedance connection and then therefore a lower quality signal. So you're less likely to be able to extrapolate information about the brain from the signal. Next slide, please. The solution that we found to scalp obstructions is creating a modular EEG. Here in the picture, we have a mannequin which has stereo EEG locations pinned to the side. you can see how if those were electrodes that were coming out of the head, you would not be able to pull a whole EEG cap over there, and you would have to rely solely on the stereo EEG data. Stereo EEG being a new neural monitoring device, could be validated using a conventional neural monitoring device such as EEG. So with this EEG, you can apply electrodes around where the large circles are, and then get as many of the 1020 electrodes as you can without putting them over the obstructions on the scalp. This also makes it easier to apply because the electrodes can be accessed from the outside of the EEG, so they can be rehydrated without removing the whole cap, removing the need for the discontinuity, or removing the discontinuity in the data over a long period of time. Next slide, please. So these are the electrodes that we have created to address that issue. They also address the issue of having a low impedance connection to the scalp. These electrodes are sponge electrodes, which means that they hold water or saline and then create a conductive interface with the silver chloride tab on the top. This tab is conventionally used with EKG and can be used for any kind of electrical monitoring of the body. So that on top of the modular EEG allows us to maintain a low impedance connection for long-term monitoring and quick apply. Next slide, please. That's all. Thank you, and I'd like to thank the entire neural web group, which is depicted on the slide.
Thank you very much. Next up, we have Chris Williams.
Hi, everyone. Is my audio quality okay?
Yep, you sound great.
All right, excellent. Next slide. Okay, so today's topic is going to be quantitative electroencephalography and quantum physics. Next slide, please. Some of the topics that we're going to be discussing today is going to be an overview of the EEG, or the electroencephalogram, and its diagnostic relevance, defining the inverse problem, and applying specific principles of physics to the EEG as it relates to volume conduction, and then we're going to conclude. Next slide, please. So the electroencephalogram is a measure of the global electrophysiological activity within the brain. When researchers such as myself are looking at an EEG, typically what we're going to do is we're going to look at a 19-channel assessment and evaluate where specific frequencies such as the alpha band, beta band, theta band, and delta band are occurring, so that we can correlate that electrophysiological activity with specific cognitive processes, behavioral manifestations, and symptomatic profiles associated with certain psychiatric disorders. So I'm aware that this is a broad neuroscience audience. So many of you are going to be much more familiar with, say, the fMRI, PET scan, and MEG. And I want to highlight that these modalities certainly have much better spatial resolution compared to the EEG, meaning that the evaluation of current source density generators is going to be much more accurate comparatively. However, these modalities are much slower than the EEG, so about 2 to 3 seconds, whereas the EEG actually detects neurooscillatory or neuromodulatory processes to the millisecond. So even though they are better at localization compared to the EEG, it is missing significant information for ascertaining the important electrophysiological generators. Typically, when we're looking at the EEG, we're going to utilize it for polysomnography studies, also known as sleep studies, to diagnose epilepsy, seizures, concussion, psychiatric disorders such as ADHD. That's recently actually been, the EEG has actually recently been approved by the APA to diagnose ADHD, and also to evaluate medication effects. One of the big topics in EEG research right now is moving towards looking at specific biomarkers or EEG phenotypes and how those relate to medication predictability as it specifically relates to ADHD. Next slide, please. So defining the inverse problem and its mathematical limitations. So when we conceptualize the inverse problem, what we're doing is we're looking at, again, that general and global electrophysiological recording, right? And thinking about those and the electrical activity across the 19 channels. But even though we know what is going on, we want to know where those generators are occurring so that we can ascertain where the dysregulation is located from according to specific regions of interest. It's important to highlight that the inverse problem has an infinite number of solutions. And some of the factors that are related to this is due to subcortical versus cortical generators, right? And we're going to get into why that's mathematically important in the upcoming slide. The EEG assumes homogeneous conductivity where it's inherently anisotropic. So when we're thinking about the EEG and the head as a volume conduction model, if we're assuming that all of the properties are uniform or are the same, this inherently, again, is going to influence mathematical estimations of current source density, because that's not true. So specific materials such as the dura, the skull, the cerebrospinal fluid have divergent conductive properties, thereby meaning there are different levels of resistance which inherently attenuate and influence the estimations of where, again, those neurooscillatory generators truly are. Next slide, please. So specific physics applications of the EEG. So when we think about the electrical signals that are occurring in the EEG, it's important to think about where they're coming from. So the neurons by which are giving rise to the electrophysiological signals, what happens is with more synchronistic firing of neurons, what happens is you get this dipole magnetic field. And when you think about the EEG electrode placement, it's not that we're measuring the electrical activity straight down, but rather we're looking at this activity and the dipolar field and measuring that from an angle. So the neurons that give rise to the electrical are actually firing at an or perpendicular angle. When we think about the EEG as a volume conduction model and we think about the electrical activity, we're modeling this as a continuous phenomenon, right, within a specific volume conduction model, again, being the head model. Some of the most important applications of physics to the EEG are actually Gauss's law and Laplacian physics, but I'm going to highlight Laplacian physics much more in this lecture because going back to this concept of the subcortical generators versus cortical generators, Laplacian physics indicate that the magnitude of the electrical force between two particles is related to the distance between those two particles. So what this means is that if you have a subcortical generator come to a cortical generator, it's going to take many more neurons to fire across a larger area of tissue for a subcortical generator to reflect the same exact signal that would be reflected if the generator was actually cortically oriented, right? And so when we think about the current source density generators, we're looking at this according to Cartesian coordinates, or also known as the Tallarac axis within, atlas within the model. And again, determining where those coordinates are inherently is going to be related to the anisotropic properties of the EEG. Next slide, please. And so in conclusion, we've discussed that the mathematical modeling and evaluation of neurooscillatory generators are largely dependent upon the synchronistic firing of the neurons, or the number of neurons which are firing, the tissue, the orientation, as well as the depth. We've indicated that the inverse problem has an infinite number of solutions as it relates to that, because you can have a similar signal reflected from a cortical generator compared to a subcortical generator. But again, the number of neurons that are going to be required and the tissue required for that will be different for subcortical versus cortical structures. We've indicated that the EEG is inherently anisotropic. And so that when we're thinking about the propagation of these electrical signals, it is not that they are propagating or being measured according to similar levels of conductivity or resistivity. And so in conclusion, what happens is what we can utilize is a software known as SW Loretta, which stands for Standardized weighted low resonance tomographic analyses. And what this does is that specific software has utilized specific mathematical algorithms to standardize the weighted configurations of the neuron matrix by which the EEG is derived from, and it actually takes into account the cytoarchitectural boundaries of the cerebrum, so that we're not just treating the tissue as taking up the entire head models, which again will yield better estimations of the electrophysiological generators. And last slide, please. And so this is the final picture. This is the picture of SW Loretta. This is an analysis that I did, and you can see that those cytoarchitectural boundaries are taken into account, and that when we're looking at the dysregulation, the numbers actually on the right-hand column indicate dysregulation according to specific Brodmann areas. Thank you.
Okay, well, thank you for that. Unfortunately, we have run out of time, so we cannot do a Q&A session, but I encourage all of the speakers to look at the questions that have been posed in the Q&A box and to please respond to it by typing. If we do not have enough time to get to them, we have copied these questions and we will send them to you so you can add your responses. I want to say a big thank you again to all of our speakers and to all of our judges today. So Dr. Christina Rodriguez, Manuel Moore, Conrad Cording, and Dr. Daniel Gonzalez. I would like to thank everyone who was watching, and please make sure that you stay tuned for our keynote talk, which is Dr. Nee Addy. So please come back at 1 p.m. for this keynote. Thank you again to everyone, and I hope you enjoyed those talks.